The cannabinoid receptor is so abundant in the brain that anyone who has any background in neuroscience will stop and say, wow.
Cannabis truly is a pharmacy in a flower.
Survival comes with cooperation.
Pleasure is healing.
You're being a warrior of the weird.
I'm Lex Pelger, Director of Education at CV Sciences, and this is the Lex Files. The endocannabinoid system is as gigantic as it is mysterious. You'd need a book as big as Moby Dick to explain everything we know about it, and everything we don't. Today's guest has been studying that system for decades, and he revels in its messy, unresolved complexity. Dr. Daniele Piomelli is a distinguished professor of anatomy and neurology at the University of California in Irvine. He's the director of the Center for the Study of Cannabis, editor-in-chief of the journal Cannabis and Cannabinoid Research, and he is one of the lions of the endocannabinoid field. Today he talks about the major endocannabinoids, the analgesic or pain-killing property of cannabinoids, how our bodies respond to the phytocannabinoids from the plant, and how genetic variants might play a role in our personalities, as well as our national characters. And before we get started, we'll define a few terms that will get thrown around. In the brain, microglial cells are a type of immune cell that acts as a first line of defense in the central nervous system. The microglial cells are key for overall brain maintenance, and they're highly influenced by the endocannabinoid system. On the neurons themselves are the presynaptic boutons and the postsynaptic spines, also known as dendritic spines. The key word here is synapse, that tiny space between neurons across which the neurotransmitters flow. And as Dr. Piomelli points out, the canonical idea in neuroscience is that information flows in one direction. Classic neurotransmitters like serotonin are released from a neuron's presynaptic bouton, which you might call their mouth, and then the neurotransmitters flow across the synapse to the next neuron's postsynaptic spine, which you might call their ears. What's special about the endocannabinoid 2-AG is that it flows backwards across the synapse and tells the upstream neuron to calm down, we hear your message, ease off a bit. It's an incredibly important negative feedback loop that keeps the brain from overexcitation. Lastly, there is PEA. That one is of special interest to me because it's in our CV Defense product. PEA is an endocannabinoid found in almost every cell type in the body, and it's another endocannabinoid with a lot of clinical research behind it. You can see more about the scientific history of PEA with a lecture by Dr. Piomelli that we link to in the episode notes, and more about the cultural and medical history with a Minute with Miles segment, also in the episode notes. So, if you want to learn about these processes in your body that are stimulated by taking cannabis, and which regulates our healthy homeostatic functioning, then you'll learn a lot from this conversation with Dr. Daniele Piomelli. Hello, everybody. I'm very pleased to be joined today by Dr. Daniele Piomelli. Thanks for joining us.
Thank you for having me.
You are renowned in the endocannabinoid science field, and I was wondering if you always wanted to be a scientist.
No, not at all. In fact, for a large part of my youth, I wanted to become an archaeologist. It just so it happens that at one point I went to school of pharmacy for some practical reasons and ended up loving the science and just stuck with it.
Did you feel like there was any similarities between your early interest in archaeology and the kind of work that you're doing today?
Yeah, I think they're conceptually the same. I used to dig out of the ground stuff that had been hidden for a couple of thousand years, and now I'm trying to dig out of nature, whatever seems important is, with respect to the endocannabinoid field. So I think it's the same mental process, really.
And so you found this love of the more biochemical part of science, and then you got to work with doctors Eric Kandel and James Schwartz, who are some of the greats of this century. What was it like to start off doing research with people like this?
Well, you have to understand that I didn't know better. I had I had done very, very little before. So this for me was normality. The personalities of Eric and Jimmy could not be more different, but they all had in common this crazy passion for neuroscience, and I shared that. So we got along pretty well. I mean, with some little bumps, but it got along real well.
And so then how did your research shift from neuroscience into more focused on endocannabinoid science?
Well, the endocannabinoids are, were born inside of neuroscience in a sense, because even though now we know that the endocannabinoid system is spread throughout the body, back then the thing you thought first when you thought about cannabinoids was the brain. And when I became involved, the cannabinoid receptor, the first one, which is now known CB1, the cannabinoid receptor was just discovered, was just cloned and identified, and people started looking at where the cannabinoid receptor was in the brain. So this is a receptor is so abundant in the brain that anyone who has any background in neuroscience will stop and say, "Wow! I mean, really is so it is so peculiar with its distribution, and it's just sheer numbers." So that's why what I did. When I thought was really an interesting pharmacology to begin with, and second is very, very peculiar localization, so abundant localization in the brain. It instantly had me hooked up and I started working on that.
What was some of your early research on this subject?
Well, I started off, you mentioned my years at Columbia University. I was working not on the endocannabinoid system, on another related signaling system in the brain. So the endocannabinoids are essentially fat-like molecules. They are, generated from the fat layer that sheaths, that covers every cell, right? The cell membrane. And I used to work on, before working with the endocannabinoids, I used to work on another fat-like molecule or lipid molecule that is involved in signaling. And my work with Jimmy and Eric was about the role of these molecules called arachidonic acid metabolites or eicosanoids in synaptic transmission. And at the time, I was using a mollusk as a model called Aplysia californica.
It's one of the really interesting things about this to me is that you're studying lipids and fats in the brain, and people often think of more traditional neurotransmitters when they think about the brain. How different is a lipid signaling system compared to some of the other ones that we might hear about more often?
Yeah, well, there is a thing in neuroscience. Neuroscientists don't really like lipids very much. We get trained on stuff like glutamate and GABA and serotonin and dopamine, and then all of a sudden here we are confronted with a signaling system that instead uses lipids, molecules that are typically associated with the structure of a membrane, right? But in reality, the last like 30 or 40 years have clearly demonstrated that lipids are really important in signaling. They differ from classical transmitters in a variety of ways. Most transmitters I know of, all of them actually, are stored in vesicles, and they are released when electric signals allow the secretion of those vesicles. So the vesicles get fused with the membrane. The transmitter is released outside the cell. That is not a mode of action that lipids can use, because lipids are because they're lipids, they can diffuse out of stuff. They can they can diffuse by lateral diffusion on the membranes. So they have a lot of more greater latitude than small molecules like glutamate or GABA do. So the way lipid signaling works is that and this is true for almost every lipid signal I know of, with few exceptions, in most cases, the lipid signal is preformed, so there is a precursor in the membrane, usually stuck to a particular place of the cell, thanks to a bunch of structures that scaffolding structures that stick it to a particular part of the cell, say the presynaptic bouton or the postsynaptic spine. So there is a precursor in there, and then receptors are activated by classical transmitters, say glutamate or acetylcholine, And these receptors, metabotropic receptors, these are receptors that activate biochemical reactions. And in this case, the biochemical reaction they activate is the activation of enzymes like phospholipases, for example, or other forms of lipases. They will cleave these preformed stored lipid storages into something that is biologically active. And this is how the two main endocannabinoids are produced, but also other lipids are produced through the same logic. It's really not something that is unique to the endocannabinoids. The endocannabinoids have brought it to a point of really high specialization, and that can be best appreciated in something called retrograde signaling, where basically the postsynaptic spine, which is at receiving end of the synaptic flow, the postsynaptic spine sends a lipid signal to the presynaptic bouton, and that lipid signal is the endocannabinoid 2-arachinyl-glycerol, or 2-AG.
And that's a very special function. The retrograde transmission. It's only that and nitric oxide that helps calm down the sending neuron.
Yeah, there are some similarities between the endocannabinoid 2-AG and nitric oxide. You brought up a very important correlation. Nitric oxide is a gas, but like the endocannabinoids, is acting very locally at probably within the diameter of one or two synapses, which is really what 2-AG can do. Cannot go very far away from the site of its production. It gets quite quickly destroyed.
And one other point about lipid signaling is it true that it's very nuanced in a sense compared to more classic signaling, where it can detect very small changes and have the cell respond accordingly?
I don't know about that. Basically every type of signaling in the brain is fascinating and different in its richness. I think what is special about lipid signaling is that it allows for fine tuning, and if that's what you meant, absolutely, it allows for fine tuning that is not impossible, but it's a little bit harder to do with, say, a peptide or a gas like nitric oxide. And the reason why I talk about fine tuning is because this is what the whole phenomenon of retrograde signaling is all about. So in classical transmission, the presynaptic bouton contains the transmitter, right? And when an action potential arrives to the presynaptic bouton, calcium gets in and neurosecretion occurs, and then the transmitter is released and activates receptors postsynaptically. So it's a very sort of structured organization with presynaptic going to postsynaptic. But the nervous system has evolved throughout the eons a lot of ways of controlling that. And the classical way is by allowing the transmitter to also act on presynaptic receptors. So the same transmitter that activates postsynaptic receptors also works presynaptically. That is very, very common. But the way the endocannabinoids work is one step further. So the transmitter again activates a receptor that releases this sort of circuit breaker, this endocannabinoid molecule, and now travels back. And someone called it a circuit breaker. Someone else a few years ago, and in a sense, it's a correct metaphor, but it doesn't always break. Sometimes it decreases the release of transmitter, so it acts as a real breaker. And in a few but important cases, it actually enhances the release of transmitter. So it's really a broad spectrum modulator. But this only works also for 2-AG. We have to remember that the endocannabinoids that we know of are two. And anandamide has one is called 2-AG, the other is called anandamide. Anandamide was the first one to be discovered. And anandamide actually has a different mode of action than 2-AG does.
Could you tell us more about anandamide and what makes it unique?
Well, anandamide is really unique because the pathway, the lipid pathway that produces anandamide, and it's only incompletely understood, that lipid pathway really is novel. When it was discovered back in 94, 95, no one believed us. what the heck is this? And instead 2AG is a little bit more standard. If you look at the textbook biochemistry of lipids, 2AG is right there, has been right there for 40, 50 years. Anandamide is also unique in that, unlike 2AG, it is able, and we don't really understand well how, but it is able to get to a greater distance from the site of its production. Than 2AG does. So 2AG stays very much, in a sort of point-to-point pattern of communication. It goes from post to pre, and that's about it. It gets removed fairly quickly. It can probably reach a glial cell nearby, maybe another synapse really nearby, but it's limited in its the extent of its spread. Anandamide is a little bit less limited, and so it moves further. And for this reason, it's more like a volume lipid modulator. So by that I mean something that spreads over a certain volume, not very big. We're talking about micrometers. We're not talking about millimeters, but maybe tens of micrometers, maybe hundreds of micrometers. But we are still talking about very small distances. But they're enough to sort of influence for anandamide to influence multiple synapses, to influence the nearby macroglia, the nearby astroglia. So in that is different from 2AG. There is also a lot more 2AG than there is anandamide, maybe 200 times more. And much of that is involved in signaling. So they're really two quite different transmitters. We're talking about the endocannabinoid system, and we think they all do one thing, but actually they divide their jobs. There is a pretty nice division of labor between these two endocannabinoids.
And so the far-reaching effects of anandamide, is that why there is a theory that there's an anandamide transporter that's kind of helping it move around, but that isn't proven yet?
So the anandamide transporter is actually probably more like transporters. There are two kinds, in my opinion. One that allows, and this is speculation, but unfortunately, we do not know really. But there must be one type that allows anandamide and allows cells to recapture anandamide. And the reason for that is not so much to reuse it, because the cells cannot reuse anandamide; they must destroy it. But it's probably to avoid a spread in places where it's not useful. So you have to appreciate that. Let me give you an example. Of if you were to use some cannabis right now, your brain would be probably stoned or high. So that's not what the endocannabinoid system does. If the endocannabinoid system was let go free, we will always be stoned or high, and maybe some of us are, but most of us are not. And so in order to avoid that, you need to keep every component of the system in place. And so the transporter is one of the things that the neurons utilize to recapture anandamide and making sure that it doesn't go in the wrong places. And by the way, probably 2AG is recaptured, though the mechanisms appear to be a little bit different. And then there might very well be proteins, we call them chaperones, proteins that bind anandamide, bind 2AG, and allow them to go at greater distances, acting like pretty much like a boat, right? In a sea of water, we imagine the cytosol of the cell, the inside of the cell, to be mostly watery, and these guys shuffle the endocannabinoids from one place to another. There is a lot of speculation about that, but we don't really know. We don't really know for sure.
And so anandamide is often called the neurotransmitter of bliss, and you mentioned how some people might be more stoned. Do you think, with the speculations of what we know about the different genetic variants of, say, our CB1 receptors and the endocannabinoid system machinery, that some people are just more naturally stoned, and that's why they act the way that they act?
Well, that's an interesting question. So first, let me say that the name, the transmitter of bliss, comes from its name itself, because when Bill Devane and Rafi Meshulam isolated anandamide back in '92, I think Bill called it anandamide because he was very fond of everything Indian and Sanskrit. He studied Sanskrit at the University of Jerusalem, and ananda in Sanskrit means bliss. So in reality, what anandamide does and what we understand anandamide does is not really to give us any sense of bliss, but rather to help us cope with really stressful situations, psychological or psychosocial stress, but also physical stress in some cases, and probably also it helps us dealing with social behavior. We are social animals, and anandamide appears to be playing a role in social reward, the pleasure that we feel when we meet, encounter, and deal with other members of our species. So it's not really the bliss molecule. So it's interesting. The genetic variance is really fascinating. There is an enzyme that this is a protein that helps destroy anandamide, and it's called fatty acid hamide hydrolase, FAAH or FA. And there is a variant, a human variant in the FA gene that contains it's slightly changed. It's really a very small change, but the protein is less functional, so it gets destroyed more quickly. And individuals who carry this variant have less Fa. So if you have less Fa, you have more anandamide, right? And so it's interesting. This is a sort of a natural experiment that allows us to ask the question, what happens if you have more anandamide? Are you high? What is the difference? And so, as you would expect from what I was telling you before, that the role of anandamide in stress and in social behavior, people who have that variant tend to have lower reaction to stress under experimental conditions. So you put them in a lab setting, and you challenge them with some slightly stressful stimulations, and they respond better than individuals who instead have normal levels of Fa, so lower levels of anandamide. So that is the suggestion is that in these individuals who have more anandamide tend to be more capable of dealing with stress, right? And it's a funny story, but there is such a thing as happiness research, and the paper came out in a journal entitled "Journal of Happiness Research," where they showed where this particular Fa SNP, this Fa variant, is localized in various nations of the world, right? So there are nations that have a higher level of this higher frequency, rather, of this particular SNP, of this particular variant, and nations who have lower. And it's funny because in Europe, for example, the nations that have the higher rate of this SNP are Scandinavian nations. They're also well known for being particularly happy. And nations instead that, like Southern European nations, who are well known for their complaining habits, have lower levels of this SNP. I mean, I say this tongue in cheek because I'm not fully convinced. These are all, of course, associational studies. They're not causative studies, but they're well done. These are well done studies, and one can sit there and look at them, and everyone can interpret them the way they want. But it's interesting that this variant should be so frequent in the population. We're talking about up to 10-15% of the population carrying this variant. So it must have some kind of adaptive value to be maybe a little bit happier, especially if the weather is not as good as in some parts of the world, right?
So it might be a little bit of a chicken or the egg.
Exactly. It gives an evolutionary advantage. I guess if you're happier, you get to meet more girls, right?
At CV Sciences, we love our full spectrum hemp products. But for some people with sensitive jobs or sensitive systems, they want something with 0% THC. So we created a line of products called Happy Lane. Made with CBD from hemp grown in Kentucky, we have gummies, we have chews, we have liquids, we have soft gels, and we have the always beloved CBD roll-ons for applying to your skin. Everything is non-GMO, vegan, purity tested, and manufactured in the United States. So when life has you wound up, tense, agitated, antsy, uptight, jittery, or jumpy, try our Happy Lane line of CBD products. For 25% off, use the coupon code LEXFILES at pluscbd.com. One thing that fascinating about what you've been saying about anandamide is how many mysteries there are still around it. There's labs like yours who've been studying it for a long time and lots of other labs in the world, and yet we still don't know the complete synthesis pathway for it. We don't know about how it gets transported around. Are there great mysteries that you would love to just be able to know that are right there, out of reach in endocannabinoid science?
Oh yeah, there are quite a few, and I don't want to bore the listener with all those, but there are really quite a few. And again, it goes to what I was saying before about neuroscientists having a problem with lipids and fat, because some of these are so obvious. One would like to understand exactly how an anandamide is produced, for example. And why? Well, because if you understand that, you understand when and where is generated for what purpose, right? And also, if you have a full understanding of all the enzymatic steps, underpinning an anandamide production, then also you can localize them in the brain. So you identify a particular enzyme, and then you go and say, okay, that's where the enzyme is. That's probably the function it's serving. We have some of that information, but we are still not there. I would say we still have about 50 to 60%, if at least based on our current knowledge, of stuff that we need to understand and understand better. And I think that has quite interesting consequences. But as I said before, I think now the field is more focused on something else, something more, I think, less scientifically interesting, but clearly more societally important, which is, can we leverage this new knowledge about the endocannabinoid system to generate medications that are helpful to the public? And of course, there is an element of scientific discovery there too, actually quite a large one, but is something that is essentially involves pharmaceutical discovery at this point. There are inhibitors for the various enzymes of endocannabinoid degradation. There are at least two of them. There are most important, PHA, I mentioned, and another one called monosaccharidyl lipase, or MGL, which works for 2-AG. And they're now excellent inhibitors for both. And the idea is that these inhibitors could be leveraged to enhance the helpful protective properties of the endocannabinoid system, for example, in pain, for example, in anxiety, and without the side effects that are caused by direct activation of cannabinoid receptors with THC. There are issues with direct activation of receptors. One of them is that, well, first of all, you activate receptors you don't want to necessarily activate. And second, when you activate the receptors, they tend to dislike it. So they tend to start acting up. And, if you activate them too much, they will shut down. Just like, a kid who doesn't like to be bothered, she will shut down, he will shut down. And so at the end of the day, you end up having the opposite result that you wanted to achieve, right? So those are interesting areas, of course, but in terms of basic science discoveries, I think we have interesting questions that really might eventually have also therapeutic applications, but they don't right now. In addition to what I mentioned before, there is the fact that an anandamide and 2-AG don't come alone. They are not, lonely signals. We have an anandamide, we have 2-AG, and then around we have the desert. No, they actually come in families, and there are multiple families of lipids that look like an anandamide or look like 2-AG, but they're not quite the same because they don't activate the cannabinoid receptor. So they don't do cannabinoid stuff, but they work where they can be produced and degraded through similar pathways. So they must be interacting, right? Maybe synergistically, which means that they cooperate for a common purpose, or maybe antagonistically, which means that one is pushing and the other one is pulling. But we don't know that. But it clearly is important to understand how these different cognate related lipid signals all work together for achieving some, presumably biological good, right? So that to me is another interesting mystery.
And it's also a great lead in to one of the areas that you know better than almost anyone else on the planet, PEA, the endocannabinoid neurotransmitter that has a lot of therapeutic potential and has been studied a lot in humans. And you were one of the early people. What first attracted you to studying PEA?
Well, thanks for asking. That's a funny story. But many years ago, a colleague and friend of mine and I were working on the effects of anandamide in pain. And back then, there were very few tools, experimental tools that we could use. Like there were not very many antagonists, etc. So, or there were no, mouse models that had no receptor, for example. Was really the beginning of this whole thing. And so we were looking for a control that we could use for anandamide. And anandamide was producing this really profound analgesic effect. And we said, okay, we're going to use something that looks like anandamide, but clearly does not activate cannabinoid receptors. And we picked palmethylethanolamide, or PEA, which you mentioned before. So PEA looks like anandamide. They have a common general structure, but PEA is different enough that it has really no affinity for the cannabinoid receptor, so it doesn't bind that receptor. And we were pretty sure that we would give it to animals and nothing would happen. And instead, very much to our surprise, the PEA was a lot more analgesic than we expected. Actually, it was exactly as analgesic as anandamide was. So that kind of put, I'm a really critical person. That I think that comes with the job. So we were doing this study, and I said, okay, stop. We can't go on. This must be something non-specific. What the heck are we looking at? Is this anesthesia? Whatever. We looked at all sort of different possibilities. There was in the early days, was 98. So I will just fast forward. Eventually, we discovered that this interesting effect of PEA, which is analgesic effect, which wasn't known before. PEA had been known for actually quite a few years, quite a few decades, primarily because as an anti-inflammatory compound. But analgesia is one thing, anti-inflammation is a completely different thing. And this effect was truly selective analgesic. So we found the receptor that is responsible for that effect, which is a strange receptor. It's not like CB1, it's a nuclear receptor. So it's a receptor that actually moves through the nucleus and regulates transcription of genes. And that receptor explains, I think, 90 to 95% of the effects of analgesic effects of PEA and also anti-inflammatory effects of PEA. That was the work of one of my grad students. So now, many years later, this was again in the 1980s, 98. Years later, PEA returned, in vogue in Europe. They started selling it as an anti-inflammatory, analgesic, with good, clinical background. It's not perfect. In fact, I don't think it's conclusive at all, but there is pretty robust evidence that PEA is indeed analgesic and anti-inflammatory. Not really good drug because it gets destroyed very quickly. It doesn't have those nice drug-like properties you like in a medicine, but is good enough for a number of inflammatory conditions, at least based on the clinical data we have so far.
What would be the most clinical, interesting applications you think for PEA?
So that is really the question, isn't it? Right. So I think where people are looking at, and this is not my opinion, I think it's the opinion of most experts in the field, they're looking at chronic pain, neuropathic pain, particularly at peripheral neuropathies, at conditions where there is either visible or invisible damage to the peripheral nerves that are painful, and conditions like this have shown in clinical trials, in conditions like this, PEA has been shown in clinical trials to be to have some efficacy. Do we have the smoking gun? No, we don't have the smoking gun yet. And one of the difficulties in working with PEA is that it's really not it's not really a very good drug. As drug medicine, we usually think as something that we give to a patient. The patient takes it, it goes into her system and distributes nicely and works for a couple of hours and then disappears, right? So that is the normal life time of a lifespan, if you wish, of a good medicine. But with PEA, it's different because it's a natural compound, right? So the body has developed all sort of ways of destroying it So we have to overcome sort of these self-destruction mechanisms. So we need to give a lot of the compound, and sometimes I think some individuals destroy it more quickly than others do, and so maybe those in which is more effective are those who are actually um, maybe less able to remove it, less able to metabolize it quickly. I don't know. I'm just speculating, but that's it's right now. Evidence for its efficacy is there. I think all in all, but it's really not, as I said, not the smoking gun. They would say, "Oh my God, clearly this is good for this, and that condition."
And on the biochemical level, is it difficult to study because it affects the nuclear receptors and can change the transcription of a bunch of genes, and all of a sudden you have a bunch more proteins in the system, and it can be hard to tell what's happening and what's doing the good?
No, that is really not a problem so much. PEA is complicated in its mechanism of action, and that's another area where we don't have full knowledge. Because, for example, PEA causes almost immediately an analgesic effect, and that it almost by definition excludes a transcriptional mechanism. Because in a transcriptional mechanism, PEA needs to travel to the nucleus, needs to activate its receptor, which is PPAR alpha, which is housed between the cytosol and the nucleus. And in the nucleus, then, turns on the transcription of certain genes, turns off the transcription of other genes, and acts in other ways to control protein expression. And those things usually take minutes, if not hours, in many cases, hours. So the immediate fast analgesic effect is mysterious. It's not unheard of because, for example, steroid hormones have also this classical, for example, estrogens have classical rapid effects, but we have not yet identified the mechanism through which this happens. It's PPAR alpha mediated, because if you remove PPAR alpha or you block PPAR alpha, the effect's gone. But how does PPAR alpha do that is unknown. So in this is complicated, but I don't think it's less complicated or more complicated rather than an anandamide. These are all complex things. Biology is not Newtonian mechanics. It's actually really complicated.
And that's one of the things you mentioned earlier I wanted to ask about, with things like THC and CBD, where we think of them acting on the endocannabinoid system, but in reality, they also affect a whole bunch of other systems like serotonin and endorphins and dopamine and glutamate. And I think it'd be hard for us lay people to understand sometimes what's mediating the positive effect of these. And so what do you think are some of the most important actions that the phytocannabinoids have in terms of their effects on us?
So that's a complex question, because phytocannabinoids there are many of them, at least 140, 145 at my last count, and they're all different. So they share a common name because they all come from the cannabis plant, but their structures are not completely different. If you look at them, you recognize resemblances, but they don't act the same way. So THC activates CB1, CB2 receptors. CBD, which is the other big guy, in the plant, does not really directly do that. They may be modulating CB1 a little bit, but most of the effects are probably carried out through other systems, other mechanisms. One thing that CBD does, for example, it blocks this enzyme PEA, which I mentioned before. So it will increase the levels of not just the endogenous cannabinoids proper, but also these other, paracannabinoid molecules like PEA and so on and so forth. So these are very complex actions, and we cannot just encapsulate them in one tiny box. So your viewers who are not specialists, I'll tell them that if you guys are confused, well, welcome to the club. It's a really difficult pharmacological question. But my views, and it's my view, is that when you look at cannabis and you think about cannabis the way we have thought about cannabis for the past quite a few years, since we started, humans started to deal with the plant, the major effects are mediated by THC. THC drives the pharmacology of cannabis. Then, so it happens that this plant is an interesting little little chemical industry is capable of generating all these interesting molecules, and some of them, like CBD, also have effects. But I'd like to remind your viewers, your listeners, that the classical cannabis, the cannabis that people, the pot and the weed that people smoked and use, that is driven by THC. You pull out THC from that, and no one would use it. It would be like, okay, some kind of oregano or sorts. Oh, it smells good, whatever. But it's not going to really affect your brain. CBD, of course, has psychoactivity. People have shown that it has effects that are, say, reducing anxiety, maybe reducing some symptoms of psychosis. These are all scattered information. Please, these are not proven facts, but there are clinical studies suggesting that what I just said is based on reality. And of course, recent studies have shown that it's anti-epileptic in children, right? Those are solid studies that have been conducted with proper controls. So there is value in CBD, but it is not that CBD is the cousin of THC. They're two different, they're distant relatives who maybe know each other, but sometimes maybe antagonize each other or collaborate with one another, but really are not working through the same system, And the same is true for anything else: cannabigerol, cannabichromine, cannabivarin, divarin, all these other. The names are all very nice, by the way. They all sound great, but we know very, very little about the way they work.
And so just two last questions then, and this will tie into what you were just saying. It's interesting you do this academic research that in one sense will never be finished and has no answers, but you also work in business and you have, I think, over 40 patents and you've started 3 different biotech companies. What's it like for you to be working in academia, but also to be working in the world of business?
To me, trying to translate all this work that we are doing in the lab, in mice, in rats, trying to translate into real, useful medical knowledge is important because we are scientists are so fortunate that we are funded to really enjoy, have fun, really, because we have fun in the work we do. So I feel that is an obligation for me to help whenever we have some technology related to the endocannabinoid system that could be potentially useful to the public out there, to try and move it there. And if you want to do that, that's something that now it's no longer academic, right? Is more industrial. So you need to take that step further. And it requires different skills. So some of us enjoy that, some of us enjoy them less. I think it's an interesting set of skills to know. I enjoy the process, but it's also very, very frustrating because unlike science, where we can control things and we can experiment, try once, twice, 3 times until we understand, and maybe it works, maybe it doesn't work, but we have a clear answer. Unlike that, the clinical world is a lot more confused. It costs a lot more, sometimes in a justified way, sometimes not. It's much more costly, and for this reason, it is also like kind of a one-shot deal. So if you say, okay, I think a phi inhibitor will work for this particular condition, you better be right, because if it doesn't work for that condition, then people, okay, it doesn't work, but it could be working for another condition, right? But you don't know that, and you will never know that because you screwed up the first time, and so there is no turning around. So it's frustrating that way, and this is a good scenario. Bad scenarios are when you have strange investors or crazy CEOs. All this stuff happens all the time. If you have a view of the market as being something rational with a lot of rational actors in it, well, I have something for you to buy. I have something I want to sell you.
And so that leads to the last question. For the future of drugs to modulate the endocannabinoid system for health, what would you be most excited about?
Well, I would be most excited about anything that works in the clinic, whatever the indication. I happen to think that conditions like anxiety, particularly social anxiety, post-traumatic stress disorder, pain are important conditions. They make the life of people who suffer from them very, very difficult. And it would be great if the endocannabinoid system helped those individuals. But I won't lie to you, if phi inhibitor turns out to be, or an MGL inhibitor or anything else turns out to be effective in any important human medical condition, I would be excited no matter what, because there is nothing like the feeling of when you worked on something for 30 years, there is nothing like the feeling that you can say, you can point to something and say, okay, this actually is helping people. And it came from, maybe directly or maybe indirectly through the work that we started, me and my colleagues started 30 years ago. I think it's a good feeling. I don't know, maybe I'm weird, but that's the way I feel about this stuff.
Well, thank you for all your work doing this research and for getting it out there in the world. And thank you for sharing with us today.
Thank you, Lex. Thank you so much.
Okay, until next time.
Absolutely.
Thanks for tuning in. To listen to other episodes, check out pluscbdoil.com, listen on all the podcast platforms, or see us on YouTube. Subscribe to the CV Sciences YouTube channel to see each new episode. And if you'd like to buy any of our fine products, use the coupon code LEXFILES for 25% off. If you have any questions, compliments, or suggestions, feel free to write me at research@cvsciences.com, and please follow the podcast on Twitter @The Lex Files Show, where I try to keep it fun. If you enjoyed this program, please give us a like on your favorite platform or share a link to your social media. It means a lot to us. The Lex Files is produced by Matt Payne. The YouTube videos are by Brendan Cleek. Thank you to Tina Molnar and Jasmine Morris for the visuals, and the music is by Jake Bradford Sharp. Our sponsor is CV Sciences, makers of America's favorite CBD oil. And I'm Lex Pelger, signing off.